Hi everyone, I'm Scott Reeves and this is a lecture for FSN 528, and this lecture is on ketone bodies, their synthesis, metabolism, and exercise performance. And here's a brief outline of what we'll go through. We're going to talk about ketone bodies in general. What are some ketogenic conditions, fatty acid oxidation and ketogenesis, because those two processes are directly related. We're going to talk about the regulation of ketogenesis, some potential detrimental effects, some potential benefits of ketogenesis, and I have some listed there. And then towards the end of this, we'll go through a overall summary. I think I'll do this in probably two parts, so this will be part 1 of 2. And one of the first questions is, what are ketone bodies? I think we've heard a lot about ketosis and ketone bodies, especially in recent years, but there is some new kind of angles and information that have been put forth in this area that has kept this as a very topical area of research. And there are some new aspects of this that are interesting, but I want to start with the basics of, what are ketone bodies. So their structures are shown here, and these are molecules that are they're organic water-soluble molecules that we can make within our body. For us, they're specifically made within the liver, and they're made within the mitochondria of the livers, and you'll see why. Essentially, they're derived from acetyl-CoA, and so that's why they're made in the mitochondria. They're produced from excess amounts of acetyl-CoA, and we see the three here, beta-hydroxybutyrate and its structure to the right. You can see it has a methyl group, a central carbon. There's 4 carbons in beta-hydroxybutyrate. And then we have acetoacetate, and acetoacetate structure is shown there. Again, another 4-carbon molecule. And then at the bottom we have acetone, which is a 3-carbon molecular structure. Now you can see that these are all generally referred to as ketone bodies, but only 2 of them actually have a ketone chemical group, and that's acetoacetate and acetone. The reason beta-hydroxybutyrate is also included is because acetoacetate and beta-hydroxybutyrate convert back and forth. So if you have beta-hydroxybutyrate, it's converted to acetoacetate before it's used in energy pathways. So they go back and forth, but beta-hydroxybutyrate has to be converted to acetoacetate before it is used for energy production as acetyl-CoA. And acetone is the least common form of these 3. It's far less common than the other 2, so it's generally not an energy source. It is slowly derived from acetoacetate, and then it tends to be expired from the individual. Okay, so these are ketone bodies. So we're going to talk a lot about them, but here's their structure. I might refer back to this. And the condition of ketosis can result when ketone bodies accumulate. And they can accumulate throughout the body, in tissues, in the blood, and then also in the urine. They can be detected in all of those. So when ketone bodies accumulate beyond normal levels, it's called ketosis. And obviously there are varying degrees of ketosis, but overall that's what it means. Now, ketoacidosis may result because 2 of the ketone bodies are acids, and they may cause the blood pH to drop. Okay, so 2 of those have that carboxylic acid group, which can affect pH. So if ketones are in the tissues, the buffering systems of our body try to control pH because of that. That can be problematic in certain conditions, but in others it's not. So what are some of the conditions that cause ketogenesis? One of the conditions is starvation. Basically, if a person is not consuming adequate amounts of calories, ketogenesis can occur. And one of the reasons is because if a person is in starvation status, they are likely not consuming adequate amounts of carbohydrate. And in those conditions, their body's fat oxidation goes up, some cases dramatically, to try and compensate for the lack of carbohydrate. So ketogenesis can result, and that can result in ketosis. More on that coming up. In diabetes, even though a person with diabetes might be consuming adequate amounts of calories, because that glucose is not able to enter into cells at adequate levels, and this is basically an untreated diabetes type 1, then that glucose is not metabolized in the cells, and that causes a significant increase in fat oxidation as an alternative energy source, and that can cause the generation of ketones. So it's often in uncontrolled diabetes, if a person is not monitoring and adjusting their insulin and glucose levels, then ketogenesis can occur and become significant. There's also a term called nutritional ketosis, and this is when a person actually manipulates their diet to cause ketosis. And the classic scenario is the high-fat, low-carbohydrate diets where the carbohydrate is significantly low, and in order to achieve ketosis, a lot of times people will eat less than 20 or 25 grams of carbohydrate a day in order to induce this ketosis. But it's because it's controlled and it's implemented on purpose. It's called nutritional ketosis. Now, there's another scenario here where if ketosis is the accumulation of ketones in the blood, generally it used to be regarded to as just because of either starvation or diabetes or lack of consumption of carbohydrates. But in fairly recent years, acute nutritional ketosis has been referred to as when an individual actually consumes ketones. They consume purified ketones, they ingest them, and that exogenous source of ketones increases their blood and tissue levels of ketones. Because you don't have to go through the typically few days or weeks that it takes to induce ketosis through dietary manipulations like lack of carbohydrate, it's an acute condition where they are immediately consumed and ketones in the blood immediately go up. So that's what we're going to refer to as acute nutritional ketosis. And this is one of the newest areas of this whole area of research. Okay, now one of the things that I mentioned here is that ketone bodies tend to be generated from an excess amount of acetyl-CoA. And this is acetyl-CoA that is in high levels in liver cells. And the major source of acetyl-CoA for those liver cells comes from the oxidation of fats. Okay? So a key component here is how hormone-sensitive lipase is activated and how that generates fatty acids that then travel to the liver from tissues like fat cells, and then how those fatty acids are metabolized to generate high amounts of acetyl-CoA. Okay, so hormone-sensitive lipase is an enzyme that's in lots of different tissues, or several different tissues, sorry, but it's in highest levels in fat cells or adipocytes. Hormone-sensitive lipase is shown by this black dot here. It is activated by phosphorylation. Once hormone-sensitive lipase is phosphorylated, it travels from the cytosol of the adipocyte to the triglyceride droplet that's stored there, and now the hormone-sensitive lipase hydrolyzes the triglycerides into free fatty acids, which run down their concentration gradient out of the adipocytes into the bloodstream. They bind hydrophobic portions of albumin, which then carry the free fatty acids to other tissues, including the liver, but also tissues like muscle cells. Okay? Hormone-sensitive lipase is regulated by hormones. In the fed state, when carbohydrates consumed, that increases insulin, and insulin decreases hormone-sensitive lipase activity. in fasting conditions or in exercise or in low or inadequate amounts of carbohydrate, the stored triglyceride tends to be used as a major energy source. Remember, our two energy sources are typically either fats or carbohydrate or a combination. If carbohydrate's not available, then we tend to use more fat for energy. Glucagon is released from pancreatic alpha cells when blood glucose is low. Epinephrine is released from the adrenal glands during physical activity, so physical stress or emotional or psychological stress. Epinephrine can go up, and those both activate hormone-sensitive lipase. So glucagon's main focus is to increase blood glucose. Epinephrine is to generate energy sources for tissues that need it. So in times of low blood glucose or exercise, free fatty acids serve as a major energy source for tissues that absorb them from the bloodstream. And this is a diagram showing hormone-sensitive lipase. So again, this is a fat cell. Fat cells or adipocytes have epinephrine receptors and glucagon receptors. So in low blood glucose and or physical activity, both of these hormones can be in the blood. They will bind to the receptors, stimulate signal cascades that stimulate the phosphorylation of hormone-sensitive lipase, activating the enzyme, causing hydrolysis of triglycerides, so the fatty acids leave those cells, bind to albumin, travel to tissues where they'll go down their concentration gradient into cells to be oxidized, typically for energy. When carbohydrate is in the blood, insulin tends to be released. The insulin inhibits hormone-sensitive lipase activity by causing its dephosphorylation. So again, when glucose is in the blood, it's typically the preferred energy source. Insulin is one of the reasons we switch from glucose to fatty acids when glucose is available. Okay, so now one of the questions is, how do we get energy from those fatty acids? Well, they are typically oxidized and they produce the reducing equivalents, FADH2 and NADH, but when they're metabolized, they also generate acetyl-CoA. So the fatty acids are oxidized, their electrons are added to FAD and NAD. Those electrons can then go to electron transport chain. Those fatty acids also generate acetyl-CoA, which is substrate for Krebs cycle. Now, per weight, fatty acid oxidation yields more ATP than glucose does. Okay? An interesting point here is that per carbon molecule, the combustion potential or the potential energy of ketone bodies is between fatty acids and glucose, because glucose typically becomes pyruvate, and pyruvate generates less energy or heat, potential energy, than ketone bodies. Okay? So ketone bodies are closer to fats, fatty acids, than they are to glucose for potential energy. Okay, but for fatty acids, beta oxidation is the major pathway of oxidizing fatty acids for energy. Beta oxidation occurs in the mitochondria, so that beta oxidation in the mitochondria generates significant amounts of acetyl-CoA in the mitochondria of whatever tissue those fatty acids are being oxidized by. Major tissues would include the liver and skeletal muscle, for example. So why is it important that it takes place in the mitochondria? Well, electron transport chain is there, and so is Krebs cycle in the mitochondrial matrix. So that's where beta oxidation occurs inside cells. Okay? Once those fatty acids enter into cells, that fatty acid has to be activated before it can be oxidized. Now, activation means adding coenzyme A, and this activation occurs in the cytosol. So we would have, for example, here's the fatty acid. At this point, we're saying it's gone into the cytosol of a cell, could be a liver cell or a muscle cell or some other tissues. Coenzyme A is added, so it becomes a fatty acyl-CoA. That requires 2 ATP to do that, and an enzyme called fatty acyl-CoA synthetase. Now this activated fatty acid has a coenzyme A attached, and that coenzyme A is essential for that fatty acid to be transferred into the mitochondria. Those fatty acids can go through the mitochondrial outer membrane, but for them to go through the mitochondrial inner membrane, they require a small little molecule called carnitine. And carnitine is a peptide that is needed to transfer the fatty acids into mitochondria. So here's our fatty acyl-CoA. So this is a fatty acid with coenzyme A attached. Coenzyme A does not go through the inner membrane of the mitochondrial structure, but an enzyme removes the coenzyme A and replaces it with carnitine. So these little carnitine fatty acids are what are called, sometimes referred to as fatty acylcarnitines, now go from one side of the inner membrane to the other side. Once that fatty acid with carnitine attached gets to the inner surface of the inner membrane, another enzyme replaces carnitine with CoA. So carnitine stays in the inner membrane. The fatty acid leaves the inner membrane, goes into the matrix, but it now contains a coenzyme A. So essentially what's happened is this fatty acid's gone through the inner membrane. It's left this CoA behind and picked up another one on the inside of the mitochondrial matrix. Okay? Now this enzyme can start fatty acid beta oxidation. We're now in the mitochondrial matrix. Okay? Now, one of the issues that can limit fatty acid beta oxidation is its transport across the inner membrane. Okay? So keep that in mind as we go forward, because that could be one of the advantages that cells have when they use ketone bodies for energy. Okay. Now, this is beta oxidation, and you've seen this before probably a few times, but here's a fatty acid that's in the matrix. This, for example, is a fatty acid that has coenzyme A attached. This one is C16. It's a saturated fatty acid, so it's palmitate or palmitoyl coenzyme A. First step of beta oxidation is an oxidation step. It generates FADH2. Then there is a hydration step. Water's added to the molecule, rearranges its structure. And now we have a second oxidation step. This time the electrons that are taken from this fatty acid are added to NAD to generate NADH. Those electrons also go to ETC, just like these did from FADH2. Now we've got electrons to support ATP production in ETC. But now, in this last step of fatty acid beta oxidation, an acetyl-CoA molecule is made because this enzyme, this thiolase, adds a coenzyme A right here, in between, basically to this carbon. And by doing so, a coenzyme A comes off. And that coenzyme A can go into Krebs cycle. It can. Doesn't always do that, but it can. The rest of the fatty acid basically repeats these steps, where it's oxidized again twice, and then another acetyl-CoA molecule is made. So for an even numbered, a fatty acid with an even number of carbons, every one of those carbons becomes acetyl-CoA. So palmitate will generate 9 acetyl-CoA molecules. Okay? And that can generate a significant amount of acetyl-CoA that a cell can potentially use for energy production. Now, here's one of the reasons carbohydrates are important, because carbohydrates can actually help oxidize fats. There's an old saying, fat burns in a carbohydrate fire, or something like that. Essentially what it means is this. Well, how does carbohydrate help burn fat? The oxidation of fatty acids can produce large amounts of acetyl-CoA. Acetyl-CoA has to combine with oxaloacetate in order to enter the Krebs cycle. So if you have a significant amount of acetyl-CoA, every one of those acetyl-CoA molecules has to combine with oxaloacetate in order to go into the Krebs cycle. Oxaloacetate is normally from glycolysis and pyruvate. If high amounts of oxaloacetate are needed, cells can get it from glycolysis. However, if oxaloacetate is low, acetyl-CoA accumulates and tends to make ketone bodies. So here's a scenario. Let's say, for example, this is the liver that has taken up a significant amount of fatty acids from the blood. This could be a condition where blood glucose is low, or during physical activity, where hormones have stimulated hormone-sensitive lipase. Those fatty acids have now gone through the blood, been taken up by liver cells, been activated, transported into the mitochondria, and now have been oxidized to generate significant amounts of acetyl-CoA. Okay? Now, these liver cells can take that acetyl-CoA, combine it with oxaloacetate if there's adequate amounts of oxaloacetate in that cell. There's a couple reasons there wouldn't be. One reason is this. Let's say, for example, blood glucose is low and glycogen levels in the liver are low. That means that glycolysis could be inhibited. Because glycolysis is inhibited, we have less pyruvate. That pyruvate could, if it was available, be converted to oxaloacetate to condense with acetyl-CoA in order to use that acetyl-CoA for Krebs cycle. So let's say carbohydrate coming in from the glucose from the blood is limited, or glycogen is limited. You essentially decrease the ability to convert pyruvate to oxaloacetate to combine with acetyl-CoA when fatty acid beta oxidation is high. There's another reason the oxaloacetate might be low. If, for example, under those similar conditions, if the liver has enhanced gluconeogenesis, this oxaloacetate is not going to be combining with acetyl-CoA. That oxaloacetate is going to be converting back to PEP so that it can become glucose through gluconeogenesis. So again, if carbohydrate intake is low, if insulin is low, then that means the gluconeogenesis is probably elevated, and that oxaloacetate is preferentially becoming part of gluconeogenesis rather than combining with acetyl-CoA. So here's the scenario. With a lot of acetyl-CoA in that liver cell, the acetyl-CoA has a few options, but one of the most common options is acetyl-CoA molecules are going to combine. They'll combine with each other and eventually become the ketone bodies. Those ketone bodies are then secreted from liver cells into the blood, and they're used by other tissues outside of the liver, mostly the brain and muscle cells. Okay? Now, the liver is doing this to provide alternative energy sources to tissues like the brain and muscle. Ketone bodies are an alternative to glucose in those tissues. Okay, remember the brain relies on glucose. When glucose isn't available, the brain cannot use fats because they don't pass through the blood-brain barrier, which is an epithelial layer that excludes fatty acids. So the brain either needs to run on glucose or ketone bodies. Ketogenesis occurs basically to provide the brain with an energy source when glucose is limiting. Okay? So the liver is providing the brain with some glucose, but as the glucose is becoming limited because, let's say, gluconeogenesis is limiting, and let's say glycogen is depleted, well, the brain needs ketones, and it relies on the liver to provide those. So under normal conditions, the brain relies exclusively on glucose. But during starvation or long fasts or limited carbohydrates, the brain can actually switch to approximately half of its energy is derived from ketones rather than glucose. So it's a significant change. Okay, so let's look at this a little bit more detail here. So here's how the ketone bodies are formed. Here's two acetyl-CoA molecules. They're combined by beta-ketothiolase. So essentially one of these coenzyme A molecules comes off, it's separated, and two of these acetyl-CoA molecules combine. That forms acetoacetyl-CoA, so we still have a CoA. But essentially we have kind of four functional carbons here. Now this enzyme, HMG-CoA synthase, adds another acetyl-CoA. So now we've added three acetyl-CoA molecules and kind of condensed them. Now we have this molecule, which is, we know it as HMG-CoA. 3-hydroxy-3-methylglutaryl-CoA, HMG-CoA. We talked about it before a couple times. We talked about like cholesterol synthesis. We know that cholesterol is made from acetyl-CoA. Well, HMG-CoA also makes ketone bodies. Okay? This enzyme, HMG-CoA lyase, splits this molecule, HMG-CoA, into an acetyl-CoA and acetoacetate. Acetoacetate is the first ketone body here. Acetoacetate can become acetone. Acetoacetate has 4 carbons, acetone has 3, and they don't show it here, but this is a spontaneous decarboxylation where the acetone is made. It's made in relatively small amounts, and once it's made, it does not go back. Okay? Acetoacetate can also become beta-hydroxybutyrate. This is 3-hydroxybutyrate. We can refer to it as beta-hydroxybutyrate. There's our hydroxyl group. Okay? Now, this enzyme is beta-hydroxybutyrate dehydrogenase. Okay? So when it goes from acetoacetate to beta-hydroxybutyrate, it's been reduced. But it can also go backwards and be oxidized. Same enzyme, goes back and forth, reverts back and forth readily. Okay, and we'll talk more about that. But in order for this to be used as energy, beta-hydroxybutyrate has to become acetoacetate first. Okay, but that's how they're made, and again, made almost exclusively in the liver. So here's another way of looking at it, simplified. Here's an acetyl-CoA molecule. Combines with another one, coenzyme A comes off, here's our acetoacetyl-CoA, remember now we're skipping our HMG-CoA, but now we go from, this is showing acetoacetate is made here, converted to acetone, beta-hydroxybutyrate, remember these are the 3 ketone bodies that we're talking about, these 2 predominate. Now the liver gets rid of them, puts them in the blood, they're non-toxic in general. They're acids, so they can be problematic, but they're water soluble. Other tissues can absorb them and revert them back to acetyl-CoA. The liver can't do this. Okay? So extrahepatic tissues can take acetoacetate, convert it back to acetoacetyl-CoA, and then that can be converted back to acetyl-CoA. So the liver's taken acetyl-CoA, made ketone bodies, put it in the blood, other tissues pick up the ketone bodies, convert it back to acetyl-CoA, and they can use it for Krebs cycle. Okay? Now, under normal conditions, those ketones in the blood are at low levels. However, as soon as a person starts fasting or induces starvation or undergoes starvation, those ketone bodies in the blood start to accumulate. These are the blood concentrations in millimolar amounts. I'll convert them to grams in a few minutes. This is weeks of starvation. Red line indicates beta-hydroxybutyrate. Blue line is acetoacetate. Okay? If a person stops consuming food, within a few days, actually overnight, these start to accumulate. Now overnight when you're fasting, these will go up a little bit, then you'll have breakfast, they'll go right back down. But if you continue to skip meals, over a period of time, you can see after a few days, they accumulate significantly, especially beta-hydroxybutyrate. Beta-hydroxybutyrate tends to be the most common form in the blood. Okay, and again, remember, these interconvert. Acetoacetate is also accumulating. So if you look at 1 week, like after a few days or up to 1 week, these are now at significant levels, and they can become a significant energy source, okay, for tissues like the brain, okay? They will eventually kind of level off, and I'll talk about why in a few minutes, okay? But again, this is kind of a way that our body can adapt to lack of carbohydrate intake, and depletion of carbohydrate stores. Our body can adapt by making ketone bodies. Okay, so let's talk about ketone body amounts and their distribution. After an overnight fast, ketone body production is occurring at a rate of approximately 35 grams a day or 24 hours. And you can see that's not a real significant amount, a little over a gram an hour is being made, right? Now, if we look at, for example, after 5 days, look how significant it is. It goes from 35 to 140 to 280 grams a day. And for example, that is now can be a significant energy source for tissues. Now, typically, that's the high. It doesn't go, it starts leveling off because of feedback mechanisms. Some of those feedback mechanisms include anti-lipolytic and insulinotropic effects. So for example, when ketone bodies start to accumulate in the blood, they'll actually inhibit hormone-sensitive lipase breaking down triglycerides. And because those fatty acids are being reduced in the blood, there's less ketone body production by the liver. Insulin, remember, inhibits hormone-sensitive lipase. So those ketone bodies, when they go up to a certain level, they can actually start stimulating the release of insulin. That's if a person releases insulin from their pancreas. Now remember, in type 1 diabetics, that's not the case. So in diabetes, a person, if they don't control their blood glucose, their blood insulin levels, then it can be a problem because they don't have some of the feedback mechanisms that people with that are not type 1 or type 2 diabetics actually have. Now, the ketone bodies can be taken up by the brain, kidneys, and skeletal muscle, among other tissues. Lots of tissues that can use ketone bodies. Now, of course, the red blood cells cannot because they don't have mitochondria, so they don't run Krebs or ETC, but most other tissues can use them. However, the brain seems to be the major target of these ketones. It tends to be better at uptake because there is a linear relationship of uptake per blood ketone body concentration. In other words, for the brain, you can see a relationship between when ketone body levels in the blood go up, the amount of ketone bodies going into the brain are in direct association with that. Other tissues like skeletal muscle, they'll tend to go up, but they tend to saturate easily. And so skeletal muscle at rest will take up only a limited amount of ketone bodies. However, one of the adaptations that tends to occur in skeletal muscle is skeletal muscle appears to easily saturate at rest, but uptake might increase during exercise. So one of the questions is, these ketogenic diets that a lot of people say help them with things like losing weight, can exercise be done on these ketogenic diets? One of the stipulations for that would be the skeletal muscle has to be able to take up ketone bodies during exercise. And it looks like there might be mechanisms where skeletal muscle actually increases their uptake of ketone bodies from the blood. Okay, a little bit more. Potential benefits of ketosis. Now, there is quite a bit of evidence to show that ketosis, or accumulation of ketone bodies, may be beneficial in persons with Alzheimer's and Parkinson's. For example, they tend to have improved, some people tend to have improved cognitive abilities with those two diseases if they're on a ketogenic diet. And also epilepsy, it tends to minimize seizures in some of the patients with epilepsy. So it's a kind of a common potential clinical intervention for those with certain types of epilepsy. Ketosis and ketogenic diets may also help in weight loss if, for example, carb intake contributes too many calories to that person's energy intake. So for some people, it can help in weight control. Again, I think one of the things that we say a lot is nutrition. A good nutrition plan is important, but it should also typically be in coordination with a good exercise regimen. But what about individuals that are exercising on ketogenic diets? Are those two things compatible? And it's not that simple, but it's getting to be tested more and more, and it's getting more and more popular. So I have a link here that you can go to this link and you can see, and this was in Cycling Weekly about a year ago, actually a little over a year ago, where they came out saying that some of the professional cyclists, even Tour de France teams, actually use ketogenic supplements. So they are actually drinking ketones, and there's some data to indicate it could be helpful to those athletes. So if you want to take a peek at that and you can get an idea of it, we'll talk about some examples of this towards the end of this lecture, probably in part 2. Okay, so those are some potential benefits of being on ketogenic diets. What are some potential detrimental effects of ketosis? Well, for example, in diabetes, in type 1 diabetes mellitus, untreated diabetes conditions, in other words, if insulin is not being used consistently or effectively, it can lead to ketosis, and that means ketones in both the blood and the urine at high levels. Now here's the problem, that those ketones are acids and they can challenge the blood pH and also the urinary pH. If there's those ketones in the blood, that can lead to sodium loss because the urine pH is going down. It increases excretion of sodium. And because of that, we can have metabolic acidosis that starts to occur. Basically, those bicarbonate buffering systems can be challenged to the point where they cannot retain a proper pH in body tissues. And because of that sodium loss, in addition to that, that can cause severe dehydration, and that can eventually lead to coma and potentially death. So diabetics need to be more careful with ketones than other people. And one of the reasons is potential dehydration and fluid losses that can occur. In other people that are not diabetic, it tends to be, I wouldn't say healthy, but nowhere near as harmful, potentially harmful. So for example, in individuals that are going through starvation but have normal insulin production and sensitivity, the ketones function as an alternate to glucose. Typically the ketones are not harmful in these individuals. The buffering systems can adjust the tissue and fluid pHs to stay at appropriate pH levels. So they tend to not be harmful in most people. Some people they can be, but now the negative effects in body composition can result especially in an initial phase of carb depletion. So if you think about it, if a person's using a ketogenic diet and they're decreasing carbohydrate intake significantly, they are essentially reducing their insulin responses. And so the individual tends to lose tissue, because if they're minimizing carbohydrate, then gluconeogenesis is increased, and most of the gluconeogenic precursors are amino acids. Most of those amino acids come from skeletal muscle that are taken to the liver for glucose production. And so it can cause significant changes in body composition because of skeletal muscle loss. So for example, and the reason that's happening is to provide glucose to the brain. So remember, the first few days of this, those skeletal muscle losses tend to be significant. So in other words, for example, they keep the brain supply with about 120 grams of glucose a day. That's a minimal amount of glucose requirements for the brain and central nervous system in most people. 120 grams. Remember, the RDA is 130. In order to do that, it takes the breakdown of 160 to 200 grams of protein from our body. And that represents about 1 kilogram of muscle per day. Remember, muscle is mostly water and other molecules, but a significant amount of it is protein. But 120 grams of pure protein from skeletal muscle represents about a kilogram. That's 2.2 pounds of muscle per day. So that person will lose 2 pounds of muscle per day in the initial stages. During that time, ketogenesis is accelerating, and that ketones can become significant enough to spare glucose requirements from the brain and central nervous system. And so that's what kind of starts sparing the muscle at that point, is that ketogenesis, and that's why it's there. And again, sometimes it can decrease glucose requirements of the brain and central nervous system to about half of the normal levels. Okay. Remember, though, you're also decreasing your ability to use insulin as an anabolic hormone for stimulating muscle protein synthesis and decreasing muscle protein breakdown. We've talked about that quite a few times. So again, especially in the initial phases, ketogenic diets are not ideal for maintaining optimal body composition, I would say. So let's look at this. So here's an overview of endogenous ketosis. So in other words, starvation, potentially diabetes, or significant curtailment of carbohydrate intake. Here's endogenous ketosis. We're going to compare that to exogenous. Okay? In the exogenous, this is when the individual is actually consuming ketones. This one is the ketones are accumulating from their own endogenous synthesis. Okay? In this scenario, remember, we're going to start over here, because in this scenario, adipose tissue is being hydrolyzed to generate free fatty acids. For example, catecholamines like epinephrine, cortisol are stimulating adipose tissue, hormone-sensitive lipase. Free fatty acids are significantly increasing in the blood. They'll travel to the liver. They generate, those fatty acids become activated, acetyl-CoA. They go through beta oxidation, generate acetyl-CoA, then acetoacetyl-CoA. Remember, they're not able to enter into Krebs cycle readily. Then HMG-CoA. Then they become beta-hydroxybutyrate, acetoacetate, which leave the liver, go into the blood. They enter into tissues through these MCTs. MCT stands for monocarboxylate transporters. So the ketone bodies go through transporters. They recognize them, transfer them into tissues. Remember, beta-hydroxybutyrate tends to be in higher amounts. Converts to acetoacetate. Then picks up a CoA, becomes acetoacetyl-CoA. And then is split to generate 2 acetyl-CoAs, and that can go into the Krebs cycle. Okay? So again, we've gone from fatty acids to acetyl-CoA molecules that are used for Krebs cycle. Okay? And this is in other tissues, including muscle. Okay? Now let's look at this, exogenous ketosis. This is, for example, when a person is consuming a nutritional ketone body or a ketone body ester. We'll talk about that in a few minutes. You're actually consuming those, so they go into the gut. They leave the gut. Remember, they're water soluble. They leave the gut. They can go through those monocarboxylate transporters. So here's beta hydroxybutyrate acetoacetate that have come from the diet directly. They go into the blood, and then once they're in the blood, they can again enter into extrahepatic tissues, and this part of it's the same. Through the monocarboxylate transporters into tissues, provide acetyl-CoA molecules for Krebs cycle, right? And remember, this could be brain, this could be skeletal muscle. Now, some of those ketones are actually going to go from the gut through the liver, and in the liver they're going to be converted, but then it's basically the same because they're going to go into the blood from that point. Now, some of these ketone bodies actually interact with this. This is a receptor that's the nicotinic acid receptor, a form of vitamin B, right? This receptor actually inhibits lipolysis. So one of the things that ketone bodies tend to do is they inhibit lipolysis. Again, this is one of the things that maybe keeps them at a certain level in the blood, is they'll actually kind of feed back and inhibit more fatty acids being released to generate more ketone bodies. How much of an issue is that is kind of still being debated at this point, okay? So this is one of the new areas of research in this, is what if we actually consume the ketone bodies rather than going through a dietary regimen that takes time and is potentially not optimal as far as body composition and other things. Why not just consume the ketones? As long as they're safe, it's an interesting angle to all of this, and I think potentially something that's going to get a lot more research, especially with all the bad press that some of the ketogenic diets actually get. Okay? Now, let's look at a couple more things here. So this is, again, metabolism in tissues of these ketone bodies. For example, here's our prolonged fasting, reduced carbohydrate availability, or ketogenic diet. We have an increase in free fatty acids here. The fatty acids go into the liver, they become acetyl-CoA, and then here's our HMG-CoA synthase, which makes our HMG-CoA. Remember, some of the acetyl-CoA becomes acetoacetyl-CoA, and then these two molecules basically can interconvert. Here's our HMG-CoA, goes through HMG-CoA lyase. Here's acetoacetate. Here's beta hydroxybutyrate. Here's our dehydrogenase. Leaves the liver through these monocarboxylate transporters, travels through the blood, and in the muscle, for example, those two molecules interconvert, become acetoacetate, and here's the thiolase that now splits it into acetoacetyl-CoA. So now our CoA has been added. Sorry, this is a transferase. I don't remember what I said. This is acetyl-CoA acetyltransferase. So now acetoacetate-CoA becomes acetyl-CoA, which goes into Krebs cycle. One of the reasons I'm showing this is these molecules here. Now, one of the limiting factors of skeletal muscle for fat oxidation tends to be how those fatty acids get across the sarcolemma and get into mitochondria. Fatty acid transport across cell membranes of skeletal muscle and across the mitochondrial membrane can be limited. So transport of fatty acids into skeletal muscle and in the mitochondria can limit fatty acid oxidation in skeletal muscle. Ketone bodies use different transporters. Doesn't tend to be limiting. So ketone bodies can provide acetyl-CoA to the skeletal muscle without the limitation of fatty acid transport. So again, it's an interesting scenario, especially if you take in those ketones from the diet, because you might be able to kind of enhance ketone body uptake in skeletal muscle and forms of acetyl-CoA in the skeletal muscle, in addition to that acetyl-CoA coming from fatty acids. So again, this is what's kind of intriguing about this. Now, if we look at it again a slightly different way here, and again, these papers are down here. This is kind of an editorial that was recently published. So now, for example, let's say we're taking in these ketone esters. They're going through the stomach, through the gut. They're absorbed in the small intestine. They go into the blood. So here, for example, is taking in a ketone ester that can actually increase beta-hydroxybutyrate. This editorial or comments, additional comments, were about a paper that used as part of its studies a ketone ester that was a form of beta-hydroxybutyrate. So the beta-hydroxybutyrate's increased in the blood by ingestion, and it's very quick. Within 30 minutes, beta-hydroxybutyrate levels go up. Now, they go through the monocarboxylate transporters, increasing beta-hydroxybutyrate in skeletal muscle, so they have data that shows this. Ingestion increased beta-hydroxybutyrate in muscles, and it's converted to acetoacetate, and then that goes through the steps and becomes acetyl-CoA. Along the way, forms of this beta-hydroxybutyrate is actually inhibiting muscle protein breakdown. So beta-hydroxybutyrate in skeletal muscle tends to inhibit muscle protein breakdown. That decreases the release of branched-chain amino acids that are typically able to become ketogenic. And they're able to contribute carbons to TCA, because remember, they can become acetyl-CoA and succinyl-CoA. The skeletal muscle seems to have a way of sensing when that's not necessary. If beta-hydroxybutyrate is there, we don't have to break down skeletal muscle to get branched-chain amino acids. So this is a potential advantage of increasing beta-hydroxybutyrate levels in skeletal muscle through ingestion. Okay? Another thing that's happening here is now we're providing substrate for Krebs cycle. We're generating more citrate. The citrate can actually come out of the mitochondrial matrix, go into the cytosol or the sarcoplasm, inhibit pyruvate kinase 1, which is the rate-limiting enzyme of glycolysis. So it's decreasing glycolysis and maximizing oxidation. If you're decreasing glycolysis and you're inhibiting glycogen breakdown, that's one of the reasons they're thinking this mechanism might be advantageous, is you're able to spare glycogen. Now the data is not all that firm on that, but it's a potential benefit of this. You're sparing glycogen. By decreasing glycolysis, you're also decreasing pyruvate. Because remember, as acetyl-CoA levels in a mitochondrial matrix go up, it tends to inhibit the pyruvate dehydrogenase complex. So acetyl-CoA is inhibiting this, which decreases conversion of pyruvate to acetyl-CoA in the matrix. So rather than becoming acetyl-CoA, the pyruvate goes to lactate. So you're inhibiting here, but you're also inhibiting glycolysis. So you're inhibiting lactate production, theoretically. And we'll look at some data that indicates that might be the case. And then over here, we have, this is intramuscular triglycerides. Remember, skeletal muscle can store some triglyceride, and it can become an energy source. Those fatty acids are activated once they're released from the triglycerides. Remember, this is in the sarcoplasm. And then they're activated, then go through carnitine. And this is the carnitine transferase here, so the fatty acid goes in for beta oxidation. So it may be a way of actually enhancing energy production from intramuscular triglycerides in this case. Okay, so we'll look at this coming up. Oh, this one's really important too. And keep this in mind. One of the other kind of potential benefits of beta-hydroxybutyrate is some of them function as signaling molecules. And so now they're looking at that it could actually affect central nervous system function and affect things like perceived rate of exertion in exercise. And that could be beneficial as well. Okay, so some of the ketone bodies, beta-hydroxybutyrate, can also function as a signaling molecule. Okay. Okay, so I think we'll stop here and we'll come back. This will be the start of part 2, and we'll look at a familiar slide, and then we'll kind of summarize potential effects of ketones on exercise, and then we'll start looking at a research paper. So I'm going to pause right there.